Welcome to the deep dive. Today, we're really going to get into the weeds, looking at how our bodies work behind the scenes. How they stay healthy, deal with damage, and even change over time. Think of it like an inside look at your own biology. We've sifted through a lot of sources, and we're aiming to pull out the really surprising stuff, the key insights for you. We'll touch on everything from, why calcium sometimes goes rogue, to how our cells fight off these dangerous things called free radicals, and even how tissues rebuild themselves. Should be fascinating. Okay, let's unpack this first one. Calcium. When we think calcium, strong bones, teeth, right? That's the main thing. But your sources point out that when calcium levels in the blood creep up too high, it can cause some really unexpected widespread issues. It's kind of like too much of a good thing, isn't it?
Yeah. It is, exactly. The condition's called hypercalcemia. We're talking serum calcium over about 10.5 milligrams per deciliter. And that extra calcium, it directly messes with how easily nerves and muscles get excited. Basically changes their normal function, the most common causes. Often it's overactive parathyroid glands, that's primary hyperparathyroidism, or certain cancers, particularly in people who are hospitalized, but other things too, like granulomatous diseases, think TB or sarcoidosis, or even just taking too much vitamin D, or certain common diuretics like thiazides.
Wow, okay, so one element, but so many potential causes and problems. And there's that classic way to remember the symptoms, right? That mnemonic: stones, bones, groans, thrones, and psychiatric overtones. Catchy. Can you break that down? What does each one actually mean for the body?
Sure. So stones, that's kidney stones. Nephrolithiasis, pretty straightforward, calcium building up. Bones refers to bone pain and sometimes even a more serious condition, osteitis fibrosis cystica, where bone gets resorbed and replaced by fibrous tissue. It weakens the bones.
Ouch.
Groans covers the abdominal stuff, pain, constipation, maybe nausea, digestive system upset. Thrones points to polyuria and polydipsia. So frequent urination and being really thirsty, it's almost like a kidney-related diabetes insipidus.
Right.
And then psychiatric overtones, that can be anything from confusion, depression, even psychosis in severe cases. And the sort of underlying reason for a lot of this is that the excess calcium stabilizes sodium channels in cell membranes, especially neurons, makes them less excitable, less likely to fire properly.
Okay, so it's affecting how cells talk to each other, essentially.
Yeah.
But it's not just floating around in the blood causing trouble, right? Your sources say it can actually deposit in tissues. And what's really interesting is there are two different ways this happens, and one doesn't even need high blood calcium.
Exactly. And that distinction is really key. We call them dystrophic and metastatic calcification. Let's start with dystrophic. This happens when calcium deposits form in tissues that are already damaged or dying. Necrotic tissue. And this can happen even when your blood calcium and phosphate levels are totally normal.
So calcium is just sticking to the damaged bits.
Pretty much. Dying cells release phospholipids and other things that calcium ions bind to, and then they form these crystalline calcium phosphate deposits. It's like mineralizing the damaged area.
Okay, so what does that actually look like? Where would we typically see this dystrophic calcification?
Right. Good question. Classic examples, things you'd see tested, include the caseous necrosis in TB granulomas, that cheesy looking dead tissue. You also see it in fat necrosis, maybe after pancreatitis or trauma, and very commonly in atherosclerotic plaques in arteries. Hardening of the arteries involves this. Damaged heart valves too, maybe from rheumatic fever years ago, or just aging.
Okay.
The big clue for you is you see calcification, but it's local, right where there's tissue damage, and the patient's calcium and phosphate labs are normal. That points strongly to dystrophic. Now, metastatic calcification, that's different. Here, calcium deposits form in normal, otherwise healthy tissues. And this happens specifically because there are high levels of calcium, or sometimes phosphate, in the blood, hypercalcemia or hyperphosphatemia.
So this is the one directly caused by the high blood levels we talked about earlier.
Exactly. These deposits tend to favor tissues that lose acid, creating a more alkaline environment locally. Think places like the stomach lining, the kidneys causing nephrocalcinosis, the lungs, and the alveolar walls, and systemic arteries.
So same causes as hypercalcemia generally. Parathyroid issues, cancer.
Precisely. Primary hyperparathyroidism, malignancy, maybe through PTHRP or bone destruction, chronic kidney failure leading to secondary hyperparathyroidism, too much vitamin D, those granulomatous diseases again. The key clue here is abnormal serum calcium or phosphate, and you see widespread calcification, often in multiple organs that were previously healthy.
That really highlights the importance of keeping that blood calcium in check. Okay, fascinating. Let's shift gears a bit. Moving from these mineral deposits to another kind of silent attack our bodies deal with constantly, free radicals. Sounds dramatic, like something out of science fiction. But it's actually a really fundamental part of how our cells work and, unfortunately, how they get damaged.
Yeah, free radical is just a term for a molecule that has an unpaired electron in its outer shell that makes it really unstable and highly reactive. It wants to grab an electron from somewhere else to become stable.
Like a tiny electron thief.
Exactly. And in stealing that electron, it damages whatever molecule it attacks. They can damage lipids, which messes up cell membranes, think lipid peroxidation. They can modify proteins, maybe inactivating important enzymes, and they can even break DNA strands, leading to mutations or triggering cell death, apoptosis.
Okay. Sounds pretty bad. Where are these things coming from? Is it mostly pollution and things like that?
Well, external factors definitely play a role, but honestly, a huge amount comes from just living. Our own bodies make them. A major source is actually our mitochondria during normal energy production oxidative phosphorylation. Sometimes electrons leak out from complexes I and III and react with oxygen to form superoxide, which is a key free radical.
So just making energy creates these damaging things.
Sort of, yeah. It's an inherent byproduct. Also, inflammation is a big one. Our immune cells, like neutrophils, actually generate an oxidative burst, a cloud of free radicals like superoxide, hydrogen peroxide, even hypochlorous acid, like bleach to kill bacteria.
Whoa.
Yeah, but that weapon can also cause collateral damage to our own tissues. And then you have environmental things like ionizing radiation, think X-rays or UV light, which can split water molecules in our cells to create highly reactive hydroxyl radicals. And interestingly, some metals that are essential for us, like iron and copper, can actually catalyze the formation of the worst kind of free radical, the hydroxyl radical, through something called the Fenton reaction, if they're not properly bound up.
Even essential minerals can turn dangerous.
In the wrong context, yes. And then certain drugs and toxins are known culprits, like carbon tetrachloride, CCLU, gets metabolized by liver enzymes into a radical that causes lipid peroxidation. Acetaminophen overdose is another classic. It produces a toxic metabolite, NAPQI, that depletes our natural antioxidant defenses.
Okay, wow. It really does sound like we're under constant attack, both from inside and out. Seems like a losing battle. But I assume we have ways to fight back.
Oh, absolutely. Our bodies wouldn't last long without a really sophisticated defense system. It's multi-layered. First, we have specific enzymes whose whole job is to neutralize these radicals. There's catalase, mainly found in peroxisomes, which takes hydrogen peroxide and breaks it down safely into water and oxygen. Very efficient.
Okay.
Then there's superoxide dismutase, or SOD. It takes that superoxide radical formed in mitochondria and converts it to hydrogen peroxide and oxygen. Interestingly, mutations in one form of SOD are linked to ALS, Lou Gehrig's disease, and glutathione peroxidase. This one's really important, especially in red blood cells. It reduces hydrogen peroxide and also lipid peroxides, using selenium as a critical cofactor. If you're selenium deficient, this enzyme doesn't work as well.
So enzymes are like the first line of defense. What else?
Then we have non-enzymatic defenses, often called scavengers or antioxidants. The antioxidant vitamins are key here. Vitamin E, that's alpha tocopherol, is fat-soluble, so it works mainly in cell membranes, stopping lipid peroxidation chains. Vitamin C, ascorbate, is water-soluble. It scavenges free radicals in the aqueous parts of the cell, and it actually helps regenerate vitamin E after it's done its job. Teamwork. Exactly. And vitamin A, or retinoids, also play a role, particularly against certain types of reactive oxygen species. And finally, remember those metals like iron and copper that could cause trouble?
Yeah, the Fenton reaction thing.
Right. So we have specific carrier proteins to keep them under control. Transferrin binds free iron in the plasma, preventing it from participating in that reaction. This is crucial. Conditions with iron overload, like hemochromatosis, lead to a lot of oxidative damage because there's too much free iron around. And ceruloplasmin handles copper and also helps oxidize iron into a less reactive form. In Wilson's disease, where you have low ceruloplasmin, copper builds up and causes oxidative injury.
It's an incredibly intricate system. Defense enzymes, scavenger vitamins, metal control. Amazing. So our bodies defend, but sometimes damage happens anyway. Which brings us to repair. When tissue gets injured, a cut, a burn, an internal injury, how does the body actually fix it? And why do some heal perfectly while others leave scars?
Right, that leads us into scar formation, the whole wound healing process. It generally happens in stages. First, there's the immediate acute phase, maybe 0 to 3 days. You get bleeding, clot formation, inflammation kicks in with neutrophils arriving first, and blood vessels become leaky.
Cleaning up the site.
Exactly. Then comes the granulation phase, say days 3 to 7 or so. Macrophages take over from neutrophils, cleaning up debris but also releasing growth factors. This is where fibroblasts migrate in and start laying down new collagen initially, mostly type 3 collagen, which is a bit weaker, more pliable. And crucially, new blood vessels start forming angiogenesis, driven by factors like VEGF and FGF. This is what gives you that pinkish, granular-looking tissue in a healing wound bed.
Okay, so building a temporary scaffold.
Sort of, yeah. Then comes the remodeling phase, starting maybe a week or two out, and this can go on for weeks or even months. This is where the magic happens, really. That initial type 3 collagen is gradually broken down by enzymes called collagenases, which need zinc to work, by the way, and replaced by much stronger type I collagen. TGF-beta is a really key signaling molecule here, driving fibroblasts to make more collagen. Over time, this leads to a mature scar. It becomes less vascular, denser, and consists primarily of that strong type I collagen, providing tensile strength back to the tissue.
So the type of injury must matter a lot then, like a clean surgical cut versus a big scrape.
Huge difference. We talk about healing by primary intention versus secondary intention. Primary intention is what happens with a clean, uninfected surgical incision where the edges are brought together, like with sutures. There's minimal tissue loss. In this case, regeneration of the original tissue often plays a bigger role than scarring, especially if the underlying basement membrane is intact. You usually end up with a minimal, fine scar. Secondary intention happens with larger wounds, big defects, ulcers, abscesses, burns where there's significant tissue loss and the edges can't be easily approximated. Here you need much more granulation tissue to fill the gap. Fibrosis, or scarring, really dominates the process. Wound contraction, where specialized myofibroblasts pull the edges inward, is also a major feature. The result is usually a much larger, more noticeable scar.
Okay, that explains different scar appearances.
Yeah.
But what about when scarring seems to go overboard, like those really raised scars or ones that keep growing?
Ah, yes, abnormal scarring. That's usually one of two things. A hypertrophic scar is when you get excessive collagen deposition, making the scar raised and thick, but it stays within the boundaries of the original wound. A keloid is different. It's more aggressive. The collagen deposition is not only excessive, but it actually grows beyond the original wound borders, invading the surrounding normal skin. They often look like firm, rubbery, sometimes itchy growths. And keloids are known to be more common in people with darker skin pigmentation, suggesting there's a genetic component to that overactive healing response.
And this whole repair process, this scarring, it's not just skin deep, right? It happens inside the body too. You mentioned heart attacks earlier.
Absolutely. That's a perfect example. Cardiac myocytes, the heart muscle cells, are considered permanent cells. Once they die, like after a heart attack, they generally can't regenerate. So the body's repair mechanism is to replace that dead muscle tissue with scar tissue dense type I collagen. This forms a fibrous scar in the heart wall. While it patches the hole, that scar tissue doesn't contract like muscle, so it can definitely impair the heart's pumping function long term. And we also see age-related changes. As people get older, there can be subtle structural remodeling in the heart. Maybe the left ventricle cavity gets a bit smaller. The septum between the ventricles can become sigmoid-shaped, and there's often an increase in collagen content, leading to fibrosis and stiffness. This doesn't always cause problems, but it can contribute to a type of heart failure called diastolic heart failure, or HFpEF, where the heart struggles to relax properly.
It's amazing how these fundamental processes, damage, repair, scarring, play out everywhere. Okay, one more area to dive into, kind of related to things depositing where they shouldn't. Let's talk about amyloidosis. This isn't just one disease, right? It's a group of conditions where proteins basically misbehave, fold wrong.
That's a good way to put it. The core problem in all amyloidosis is the deposition of abnormally folded proteins outside of cells, in the extracellular space. These misfolded proteins aggregate into insoluble fibrils that are very stable and resistant to breakdown. On a biopsy slide stained with standard H&E, they often just look like amorphous, pink, glassy material. But the key diagnostic feature, the thing that really defines amyloid, is how it looks after staining with Congo red dye and viewing under polarized light. It shows this characteristic apple-green birefringence. That specific optical property tells you it has that abnormal beta-pleated sheet structure, regardless of which protein it actually is.
Apple-green birefringence.
I got it.
And you said it's not just one protein, so different types of amyloidosis exist.
Exactly. There are several important types, classified based on the specific precursor protein that's misfolding and depositing, and each type tends to affect different organs and have different clinical associations.
Okay, can you give some examples?
Sure. Probably the most common systemic form is primary amyloidosis, or AL amyloidosis. amyloidosis. Here, the precursor protein is an immunoglobulin light chain produced by abnormal plasma cells, often in the context of multiple myeloma or a related condition. AL amyloid can deposit almost anywhere, but classic targets are the kidneys, often causing nephrotic syndrome and kidney failure. The heart is another big one, leading to restrictive cardiomyopathy. The heart walls get stiff and thick. It can also affect the GI tract, nerves, skin, and even cause an enlarged tongue, macroglossia.
Wow, virus systemic.
Then there's secondary amyloidosis, or AA amyloidosis. This develops as a complication of chronic inflammatory conditions, things like rheumatoid arthritis, inflammatory bowel disease, chronic infections like TB or osteomyelitis. In this case, the precursor protein is serum amyloid A, SAA, which is an acute phase reactant that's overproduced during chronic inflammation. AA amyloid tends to deposit mainly in the kidneys, liver, and spleen.
So inflammation drives this one.
Right. Then there's age-related or senile systemic amyloidosis. This becomes increasingly common in older individuals, usually over 70 or 80. The protein here is actually a normal protein called transthyretin, or TTR, specifically the wild-type non-mutated form. For reasons we don't fully understand, this normal TTR can misfold with age and deposit primarily in the heart, also causing restrictive cardiomyopathy. It's actually a very underdiagnosed cause of heart failure in the elderly.
Interesting. So just getting older can predispose you to this.
It seems so for the TTR type. And finally, a really famous example of localized amyloidosis is Alzheimer's disease. Here, the amyloid protein is A-amyloid, which is derived from a larger protein called amyloid precursor protein, APP. These AP deposits form the characteristic plaques in the brain tissue, contributing to the neuronal dysfunction, memory loss, and cognitive decline we see in Alzheimer's.
So Alzheimer's is technically a form of amyloidosis, just localized to the brain.
Correct. Different protein, different location, but the same underlying principle of misfolded protein aggregation into beta-pleated sheet fibrils.
It's fascinating how one basic mechanism, protein misfolding, can lead to such vastly different diseases, depending on the protein involved and where it ends up. Kidney failure, heart failure, dementia.
Absolutely. It really underscores the importance of proper protein folding and clearance mechanisms in maintaining health.
Wow. Okay. We've covered a lot of ground today. We went from calcium, not just being about bones, but causing stones, groans, and deposits.
Right, distinguishing between dystrophic and metastatic calcification.
To the constant battle against free radicals generated by our own metabolism and the environment, and the amazing antioxidant defenses we have.
Enzymes, vitamins, metal carriers, the whole system.
Then we looked at how tissues repair themselves, the whole process of granulation tissue and collagen remodeling leading to scars.
And how that can sometimes go wrong with hypertrophic scars or keloids, or lead to fibrosis in internal organs like the heart.
And finally, this whole complex world of amyloidosis, where misfolded proteins clump together and cause trouble, from systemic diseases to Alzheimer's.
Yeah, what really stands out, I think, is how interconnected everything is at the cellular and tissue level. A small problem in one area, like maybe handling iron poorly, can lead to a cascade of oxidative stress and damage. Or chronic inflammation, meant to protect, can end up causing secondary amyloidosis or excessive scarring. It's all linked.
So for everyone listening, what's the big takeaway? I mean, it's a real reminder of the incredible, constant, mostly invisible work going on inside us, isn't it? It highlights how complex health really is, how delicate the balance can be, and maybe why sometimes small disruptions can have surprisingly big consequences down the line.
Exactly. Understanding these basic processes of damage, defense, and repair gives you a much deeper appreciation for physiology, both in health and when things go wrong in disease.
So maybe next time you feel a little ache or notice a cut healing, just take a second to think about the incredibly intricate biological symphony playing out underneath. And it makes you wonder, what other hidden processes, what other subtle cellular dramas are shaping our health in ways we're only just beginning to understand? Something to think about.